Thank you for joining us for this IMeRG inaugural seminar. This came out of discussions with Charles and I, basically about all the people we want to hear talk. So Hannah was the first on the list. So yeah, really looking forward to hearing a talk about replication studies and how they're important in ecology. Thanks, Hannah.
Thanks, Matt. I'm honoured to be the first speaker in this series. I think it's going to be a really exciting series. I'm mostly excited about the following weeks, so I'm glad to get this out of the way as well. So I'm going to talk a bit about replication and how I understand it as fitting in with the way we do research in ecology. So when you're thinking about replication, you might be imagining this definition of replication, which I'm going to sort of call the original interpretation, which is often known as direct or exact replication. Essentially, it's repeating the same study. Usually it's used in the context of experiments in the same environment with the same population, same interventions, same analysis, and usually they do it with an increased sample size because there's a concern that a lot of the literature doesn't have enough sample size to reliably find an effect, and therefore if you replicate the study you might not find the same effect, but it doesn't necessarily tell you anything about whether that effect is a real relationship in nature. It might just be telling you more about how underpowered the original study is. So the idea often is to increase the sample size in the replication. And this kind of study tells us about whether the result is affected by sampling error, whether it's likely to be influenced by questionable research practices or mistakes that you might have done, the original authors might have done in their analysis or approach the first time round. And it can also turn up fraudulent data if you redo the study and find out that you can't find the same result or anything like it. It might be fraud. Hard to tell though unless you dig into that a little bit further. Ooh, I've got an echo. Sorry about that. So there's been a number of large-scale projects in other fields outside ecology where they redo studies in the spirit of that original definition of replication. So they do them as closely as they possibly can to what the original study did. All of these studies here, except for that one, the second column, take this definition of replication more or less. And you can see that when they repeated these studies, in some case hundreds of studies, in some case not quite as many, they found the same results as in the original studies less than 50% of the time, except for an experimental philosophy, which is an interesting niche case. And I've got some theories about why that's happening, but I won't go into them yet. I can talk about them later if you like. The second column there is a study by Begley and Ellis, which I like to call a corrected replication, where they've gone through and corrected the methodological problems in the studies before they replicated them, which accounts for why they've got such a much lower rate of replication, successful replication, than any of the other studies, which is also deeply worrying for other reasons, but that's outside the scope of this talk. So part of the reason why the replication rate, the rate of successful replication is so low is because we're doing a number of practices as part of our typical research practice that is driving an increased number of false results into the literature. So these are things that broadly break down into these three categories. There's p-hacking, where you're essentially doing things to your data that make it more likely to show a cool and interesting or significant result than it would otherwise be. This might be something like checking how your results are looking as you're collecting data and then stopping as soon as your result is either statistically significant, or if you're not working in a framework that looks at statistical significance, whether when the relationship or difference is strong enough that you think you can make a good story about it. It's the same kind of idea, same pressure, it just looks slightly different. It also can refer to when you decide how to include or exclude data points like outliers. If you do this after you look at your data and have a look at whether that's going to impact on how cool and notable the result you get is, and before you decide to make that decision, it can inflate your chances of finding results that are driven by that choice rather than driven by any kind of true underlying ecological relationship. Then we've got cherry picking, which is where you do a range of different analyses and then you end up only reporting the ones that look statistically significant or cool and noteworthy, depending on your framework. This increases the chance that you're going to be reporting something that's a statistical artifact, essentially. So if you do a whole lot of tests, you're much more likely to find something, regardless of whether it's there or not. And unless you report all the tests that you actually did, nobody can have any idea about how many tests are underlying that, and therefore how likely it is that that one single result that turned out cool or statistically significant is to be a true representation of nature rather than a statistical artifact. And that kind of cherry picking work comes together with hypothesizing after the results are known, which we call harking, which essentially is when you, well, it can happen two ways. Either you've done a whole lot of analyses, find one cool thing, and then you write up your study as if that's what you'd always intended to find. Or it might mean that you didn't actually, so you might have had a hypothesis and ended up not finding it and finding something else, or you might have just not really had a hypothesis, which I think is often the case in ecological studies. You just sort of go out to see what's going on. And then because of the way people want journal articles to appear and what's acceptable at journals, we end up having to shoehorn that exploratory work into a implied a priori hypothesis framework, which is misleading and very unfortunate, I think. So we did a study on questionable research practices where we asked ecologists how often not how often. So we asked them whether they'd done these practices at least once, which is the same as two studies that looked at the same thing in psychology. And subsequently, there's been another study that looks at the same thing in education. So we've got a nice cross-disciplinary comparison of questionable research practice rates. And you can see that these top ones here correspond to this cherry picking idea of doing lots of analyses and only reporting the cool ones. And that firstly, the rates of this in ecology are pretty similar to in other fields, where there is known to be a problem with replication success. And secondly, they're really high. So it's about 50% of people or more that have engaged in some of these questionable research practices at least once. Similarly for harking, interestingly here, it looks like ecology is harking more, so that's hypothesizing after the results are known, more than the other fields that we've got data on, only a little bit more than education. I suspect that's not a meaningful difference. But I think this is to do with how we write our articles in ecology. In psychology, often there's an explicit expectation that you're going to outline your hypotheses as such and say them explicitly. Whereas in ecology, in my experience at least, it's more the case that you write a story about how the information in the literature means that it was sensible to do this study in the first place, that implies that you had a hypothesis to start with, but it's rarely overtly said. And I think that distinction between having to overtly say, this is my hypothesis, when that's not necessarily true, whereas ecologists and maybe people in education are more narratively implying a hypothesis accounts for that difference. I think if you made people say their hypothesis explicitly, they'd be less likely to say something that wasn't true than if they just had to sort of narratively imply it. And then for p-hacking things, we're looking at, again, reasonably high rates of these things. So about 1/4 to 1/3 of people have done at least one of these activities. So data exclusion after results are known. I think the result here is interesting because in ecology and also education, the rates are substantially lower than in psychology. So I think that this is actually, again, driven by the necessities of ecological work. Because we spend so much time out in the field collecting our data, each data point is precious. And we're very loath to get rid of even a single one, even for good reasons, which is why I think our rates of excluding data after results are known is lower than in psychology, where often they might be doing a study where they do an online survey and each individual data point isn't as valuable to them as each ecological data point is, I think, some of the time at least. But aside from that difference here, and a lower rate of data peaking, which is to say checking the results before you finish collecting your data, I think that that's also probably driven by the way we do our studies. We'll probably go out and there'll be a set number of sites that we can have or animals we can catch, and we'll catch all of them. And that's not really a flexible number so much of the time. Whereas again, if you're doing more of like an online survey or something, it's more flexible to collecting more or less data. Which is to say, I strongly believe that the only differences between fields and these rates of questionable research practices are driven by the logistical requirements of the research in those fields and not by any kind of underlying differences in the culture between between people in different fields of research or differences in their desire to engage in or not engage in these questionable research practices. I think that very much it's reasonably the same across all fields of science because we're all experiencing the same pressures. And therefore, these low rates of replication success that I showed you in that graph before in other fields are extremely worrying for ecology, even though we haven't been able to or haven't actually done a large-scale replication project in ecology yet. So replication studies can help us know when questionable research practices have occurred, but how often are we actually repeating our studies? So there's been a few studies by people, often by Matt Markle, he did a couple of these, to look at what proportion of the literature contains studies that identify themselves as replications. So in psychology in 2012, he found that this was 1%. It's probably gone up a little bit by now, but it might be still a similarly low percent if you exclude those large-scale replication projects. In education, it's even lower at 0.1%. And in marketing research, it's a little higher at 1.2%, which is still really low. Last year, Clint Kelly brought out a paper about the rate of replication studies in ecology, and he found that there's 0.02% of ecological studies identify themselves as replication studies, which is obviously really low. But this is partly driven by the fact that it's replication studies aren't really in the ecological consciousness, I don't think, at the moment. And there might be studies that have elements of exploring the reliability and validity of other people's findings that don't identify themselves as replications that are in the literature. And Clint Kelly, again, in 2006, he did some work on this where he looked at a couple of issues of these 3 journals and identified manually whether he thought that each of the studies in it constituted a replication of these three different types. So he had direct or exact replications, which conforms to that definition I went through before. Conceptual replications, where some elements of that original study are deliberately altered. And then quasi-replications, where it's a similar study conducted in a different species or study system. I think in this case, it probably would have exclusively been species because he's looking at behavioral ecology research, but it would be equally applicable if you're studying, say, one ecosystem, substituting it for another similar ecosystem. And this sort of is an interesting question about what constitutes a replication study and what it means, particularly seeing as that original definition doesn't really fit well with ecology because of the strong interdependence between what results we get and the environmental conditions and on the day, on the year, in the season when we collect those results. So this is a table that's come from a study that I'm hoping will come out in Ecology and Evolution any day now. It's currently available as a preprint. It's just a little ad. And it's looking at different types of replication different types of replication studies and what they tell us. So broadly speaking, when I'm thinking about a replication project, I'm thinking about studies that are sort of covered in this table. So on the top row, that's the direct replication idea I told you about before. So it's a study in the same location with the same environmental conditions in the same study system, looking at the same variables and doing the same analyses. You might also do a similar study where you alter things like location, environmental condition, variables, and even study system if it's within the scope of the original study. So say I was doing, I'll pick this apart even further in a little bit, but say I was doing a study on the relationship between tree height and altitude, and I made a claim like for every 100 meters of altitude, trees are 10% smaller. And I didn't specify that that claim was only restricted to the location I sampled it in or the study species I was looking at. It would be reasonable, I believe, to test that study in a replication in any location, in any species, if it came within a similar altitude range, because that's the only thing that's specified in that specification of a study scope. From that conceptualization, a replication is designed to test how reliable the findings are within the scope of what the original study claimed. Which seems a bit silly, really, because if you made all of the exact same changes, but they weren't within the scope that that original study specified, it wouldn't necessarily qualify as a replication, even if the difference there is just a small difference in the way that author decided to express what the scope of their study is in the instant that they were writing it up, or possibly what some reviewer suggested at the last minute after 5 rounds of revisions. Because these tests that are outside the scope are more about how generalizable the findings are outside the scope of what the original study claimed. But the problem here is what's within the scope. We've got a little project going on at the moment to look at this, and I'll tell you a little bit more about it later. But the scope of studies is sometimes overstated. This is a quote we pulled out of one of the papers we were looking at. It says, "These techniques are not only applicable to identifying species from eDNA deposited on snow, but can also be used to identify species from a variety of potential DNA sources, hair, saliva, blood, urine, scats, across any medium, species, or landscape." which suggests that I could repeat that study in any medium, species, or landscape, looking at hair, saliva, blood, urine, scats, or snow, and consider that a test, a replication of that original study. The best specification, or should I say the narrowest specification, maybe they really truly believe that that would be the case, that the results would hold true in all of those circumstances. I can't know what their beliefs are. The most specific one we came across is this one. We show that attempts to improve habitat on public lands via grazing restrictions could result in greater system-wide fragmentation of sage-grouse habitat from unintended habitat loss on private lands. And it goes on to say, other sagebush-associated species could be affected by grazing restrictions on public lands. So it's basically saying, I'm making this specific claim about sage-grouse in this specific context, but you might also find similar results for other species of this type. These are two reasonably extreme examples. Much more commonly, the scope's unclear, and there are a variety of statements scattered throughout the paper which variously imply larger and smaller scopes, and no cohesive statement of exactly where and when you'd expect this result to hold true and where you'd expect it not to hold true because of legitimate differences in the context or environment. To pick this apart a little bit more, here's a nice example replication study. Say there was a study on the effect of runoff on coral bleaching. One version of that study might make a general statement about the effect of runoff on coral reefs. A different version of the same study could have equally said that that study is only relevant to the Great Barrier Reef. If you're in the world on the left and you repeat the study in Hawaii using the same methods at the same time of year, or even theoretically at a different time of year, that would be a test of the reliability of that claim. But if you happen to end up in that second world where they said something more specific, it becomes the same exact study becomes a test of generalizability rather than a test of reliability. But do the people on the orange side actually really believe that the results would be relevant to all coral reefs? It's hard to know. And on the right side, did the researchers really only believe that the results would only be applicable to this reef system? Or did they actually have good reasons to think that it might be applicable more broadly? Without directly asking them, it's impossible to know, because I believe so little thought goes into what we're actually specifically saying about how far we expect our results to extend. So in a world where it's very unclear what people meant by the scope of their original study, it leads us to this new wave of understanding of what a replication study is, which is this idea that anything where you repeat the study, but you're only changing things that could reasonably believe to influence the finding, is a replication study. I think this is really good because it allows meaningful replication studies regardless of how well the scope of the original study is described, and it's realistic given the state of the current literature. But it's based on the replicator's judgment of what's a reasonable change, and that's going to be open to a whole lot of criticism about what is a reasonable test of the original study. Even in the context of existing replication projects where they've tried really hard to be as close as they possibly can to the original studies, they're littered with criticisms about how there was underlying moderators that they didn't account for, which had changed between the original study and this study. And I think another problem with this approach, and maybe this is a little bit too philosophical or idealistic, is that it doesn't encourage people to get better at specifying what their study scope is. And I think that that's a real problem. Particularly in ecology, I think it's really important that we pay more attention to that and get better at it. both because it's an easier way to understand what a fair test of a finding is, and also because without being really clear about what your expectations for your generalizability and reliability of your findings are, it's hard for people to know whether they can actually apply them to any given management context. It's particularly important if you're interested at all in doing applied research. If you're just putting something out there with a really general statement, or even with a really specific statement, when it could be applied, when the findings could actually apply beyond that, you're limiting the effectiveness of your research, or you're being misleading and which could potentially result in suboptimal management decisions. Also, from just a purely fun research perspective, if we identify the scope of our studies more accurately, it helps us identify future areas of research. So if you say, my study will apply in X and Y case, but probably not in Z case, there's a whole lot of explicitly identified studies there where you could investigate the limits of the reliability and generalizability of that particular finding. So I think I'm making a case about why replication studies are important, but the reality of the situation is that there's really not very many of them. And we asked researchers, ecologists, why this might be the case, what obstacles there are to replication studies. And there was a few different themes, and these are three that I think were particularly common and relevant. Firstly, there's a problem with academic culture. One person said, I think most scientists want to be known for original work, not doing someone else's science. There's also difficulty funding and publishing replication studies. Someone said, it's hard to publish, very limited resources for biodiversity or ecology research anyway. So in a world where there's not very much resources, it's hard to justify using them on replication studies. And then this last point is sort of the whole reason I'm giving this talk, because there's a lot of skepticism, I think, among ecologists about whether replications are worthwhile. And this is a good example quote of that. It's really difficult in ecology to actually replicate a study. Conditions are always changing, so if you get a different result, are you really refuting the original study? And I think this is something that I hear almost every time I bring up replications to ecologists. And it's a tricky problem. I think I've tried to get at it with this idea of the study scope, but it's really tricky because, as I said, the scope of studies is very underspecified, and it's hard to tell what original authors believed or think is true. But something that I keep coming back to is, if we truly believe that our results are not replicable under any circumstances, that they're only true for the instant in which we surveyed them, why are we writing them up? Like, why are we reading them? What use are they to the world, to nature, to conservation, if they're not relevant beyond that very specific context? And I don't think it's realistic. Unless you're studying a one-off phenomenon, which is a totally legitimate thing to be doing, there is going to be a broader application of any study you do. And you, the person who did that study, are the best person to know what limitations there should be upon that, even if it's just a judgment call. All right, so that's the end of my pontification about replication studies. I thought I might just tell you a little bit about a few things that I've got in the works, because this feels more like a lab meeting than a formal presentation. So this is all works in progress. So there's 3 projects I'm particularly excited about at the moment. One is about specifying study scope. One is a many analysts project, which a few of you might be involved in. And the other one is one that I've recently applied for a grant for, which is to do with starting a replication project in ecology. So for the study on specifying study scope, we've I got a team of researchers to extract statements about the scopes of studies from 60 different articles and look at where they occur in the article, how general they are, and we're going to try and bring our thoughts about this together to provide advice about how to do that better. And the centerpiece of that is going to be a before and after look at the scope that we've specified in one of our previous studies. We're going to hopefully find a really terrible one of our own and pick it apart and show how we would ideally have liked to rewrite that if we had another chance. To hopefully both really signpost the fact that this is something we all do terribly, even if we think that we might be able to provide some advice about how it might be done better. Then there's the Mini Analyst project. This is extremely exciting, and I've been meaning to send a update to the team. Well, I meant to send it last week, and then it got caught up in a workshop cyclone, but I'll be sending that this week, hopefully. The idea here is that we're going to get many people to analyze the same data for the same questions. Because often in science, I guess, broadly, also in ecology, you see one result in a paper, and that result becomes the emblematic answer to that question in that context. But the reality is, it's possible that a different person would have had a different answer because they had a different approach. And we're not sure how often that's the case or how different these results would be. And the idea of the project is try and get at that a bit, to look at how differently people approach the same data sets and how different the results turn out as a result. People have been wildly enthusiastic about this. I've had lots of emails from people saying, I've wanted to do a study on this for so long, which just means I think it's the best time to have jumped on it. 585 people have signed up to the project. 245 of them have completed their intake survey, where I'm just collecting basic data on who they are and what their background is. 212 people have downloaded their Eucalypt dataset. So there's two datasets we're looking at. One of them's a community ecology piece with Eucalypt data, and the other is a behavioral ecology piece with blue tip data, and that's been downloaded 148 times. And 34 people have already finished their analyses. We're hoping to be, best case scenario, finishing up the analyses towards the end of June. So the fact that 34 people have already done it so far is completely amazing and overwhelming. On to the very last slide, very last project. This is the apple of my eye, my one true love, the Ecology Replication project. The idea here is to select some studies from the ecology literature. I'm hoping to do some kind of representative sample rather than picking high profile ones, so that we can make a more general statement about the state of ecology literature, and collect existing data sets that use the same variables. I've observed that a lot of the time, particularly in some areas of ecology, we collect very similar data on very similar things, and we could easily use those different data sets and capitalize on the fact that we're collecting data often from variables that we're not actually that interested in just because it's common practice, to really get an idea of how far the findings from those original studies will extend. So we want to collect a bunch of data sets that you could use to test the same findings from the original studies. We want to ask the original authors which data sets out of those they'd expect the findings to replicate in, to try and overcome this poorly specified original study scope problem. And then we'll run the original analyses on the different data sets to try and work out how well original authors can actually predict their study scope when they're really trying. And then also what the limits of the reliability and generalizability of the findings of the original study are. So does it hold true only in these contexts and not in those contexts? Was it a thing that was only, as far as we can tell, only true of that instant in time? I think that it's going to be a really interesting way to examine replication in an ecological context, because there's a lot of, it's a huge grayscale of differences between different studies. So if you did the same study the very next season, you might get a different response because there's been a flood, or there's a drought, or someone cut down the hollow bearing tree in the next paddock. But if we look at it in a variety of different data sets, it should give us an overall picture about the limits on the findings of those studies. All right, that's it from me. I'd love to hear any questions or anything you might have.
I'm interested in the obstacles to replication in ecology. When people say it just can't be done, and I know Matt's mentioned this before, but I'd just love to hear from both of you on a bit more detail about why ecologists feel like their study is so special, you know, because as a statistician, I sort of see, say, you know, a regression model and think, well, this is something Matt and I were talking about a couple of months ago. Regression model, it's you know, it's a similar kind of setup, the model that they're using, but they will insist that no, in this case, it's completely different. But I, as a math stats data person, I don't have the domain knowledge. I'd be interested to hear more about that.
I think there's a few things going on there. One, I think, is that a lot of the time, really solid ecological studies are collected, the data is collected over like 20 years or something, and that's just logistically problematic to repeat. Or even in the case that I'm talking about with substituting data, there's just going to be a limited number of long-term data sets that you could use for that kind of thing, which is problematic. I also think that a lot of ecologists come from sort of a naturalist perspective, where they're very deeply connected to the specifics of the environment that they collected things in. And I think that can sometimes blind people about the potential for generalizability beyond that specific context. Do you have thoughts, Matt?
Yeah, I think I'd agree with that. Ecology is complex, and you know, but so is everything else. We have this belief that ecology is special. We don't realize that medicine, for example, is equally as bearable and difficult to pin down. It's just that we see that and work with it every single day. So it's yeah, I think that's probably the main thing. I struggle. I think your way of thinking about the scope really helps, because I struggle with this. I did a talk a few weeks ago about recent paper, and my boss was saying exactly the same thing. You can't do replication in ecology. It's impossible, because one animal does one thing, and then it's of the same species, another individual does something completely different. And I couldn't really find a way to actually back up my argument well enough. But thinking about the scope really would help, actually.
It's a bit of a tricky minefield, isn't it?
Yeah. So any more questions? I've got a question if no one else has. So the sort of stuff that we do at NINA is very much very applied ecology. So we work very closely with managers, and we try to answer management questions. Obviously, budgets are limited, time's limited. How do you think we could possibly bring replication ideas into our work to make it more robust?
Yeah, I guess that's a really good question. So have you heard of conservation? I think it's called Conservation Evidence. It's a blog you know about it, where they try and compile evidence for the effects of specific management actions across multiple studies, which is basically a pre-compiled meta-analysis approach, which does get at things to some extent. It's completely limited by the fact that a bunch of results are never going to make it into the literature due to publication bias, though, which is problematic. I think that I don't know whether you've got similar problems to public service places that I've encountered. I don't want to name any names, but there's often a difficulty with the way data is stored, which means that data that's been previously collected for similar projects isn't utilized optimally. And I wonder whether there's something in that space where you could look at data that's been collected in similar contexts by the organization in the past and leverage that to get a broader idea than you'd have from the specific season that you're looking at. Beyond that, I think it'd have to come down to calling out to people you know are working on similar things and asking them to share their data so you can use it for your analyses. Because just repeating a whole study in real time isn't really feasible in an applied context like that, I don't think.
Thank you. And thank you guys for all you guys coming